We have a good number, and great. So welcome everyone. It's my sincere and deep pleasure to host today Professor Kelly Lambert. Professor Kelly is a dear friend. We spent almost 2 years together during COVID-19 at the University of Richmond. So I had the pleasure of knowing her very well and visiting her house and learning about her research, which is fascinating, by the way, because she marries between basic biology, the real world outside, and everything in between, and things that matter to us in our everyday lives. And she brings to life, nature around us. We tend to forget we're in our rooms, in our homes, in our houses, and we only focus on humans, yet we can learn so much from nature around us and the animals around us. So she's going to fascinate us with her talk. But first, let me introduce her. Professor Kelly Lambert is the Trawick Professor of Behavioral Neuroscience and Psychology and coordinator of the Neuroscience Program at the University of Richmond, which is in Virginia, United States. She received her Ph.D. in Biopsychology from the University of Georgia and enjoys teaching courses related to neuroscience, behavioral processes, and evidence-based methodologies. By the way, guys, she has a lab and she has all these undergraduate students who have the most fantastic time with her working with rats. Her research focuses on experience-based neuroplasticity, which she's going to talk about, using rodent, raccoon, and non-human primate models. She has received multiple teaching awards, including the Virginia Professor of the Year and the State Council of Higher Education Outstanding Professor Award. Additionally, she is the recipient of the 2018 Outstanding Achievement Award given by the International Behavioral Neuroscience Society for her research accomplishments. Dr. Lambert not only has written textbooks, but she has written 3 mainstream books, meaning for the general public, which we are going to hopefully recommend for you to read. Lifting Depression, a neuroscientist's hands-on approach to activating your brain's healing power, and the Lab Rat Chronicles, a neuroscientist reveals life lessons from the planet's most successful mammals. And most recently, which I love, is Well Grounded, the Neurobiology of Rational Decisions. And she's authored multiple, over 75 scholarly articles, and has written popular scientific articles in popular magazines like Scientific American, the New York Post, and the Chronicles of Higher Education. And she has been interviewed by many radio stations, and she has her own YouTube video series, and she has a TED Talk, which is amazing as well. So we're going to share all these links in this talk, and later I will share it to the rest of my students as well. So we are very, very happy to have you with us and to give us from your time and from across the oceans. So the floor is yours. And just to give people a general overview, Kelly's going to talk for 45 minutes, and then we will open the floor for questions from the audience. You can either write your questions in the chat, and you can keep writing even while she's speaking. And then, and we can open the floor. You can raise your hand and actually speak up, open your camera, introduce yourself, and ask any question to Kelly. And we will end at the top of the hour. This is being recorded. So the floor is yours, Kelly. Thank you very much.
Great. Thank you so much. You guys are so lucky to have Professor Dejani as your professor, and we miss her so much here. And it was a challenge during COVID, but we made them the most of what we could do here. So it's a real treat to be here with you today. And I'm probably going to cover too much because I get so obsessed with what we're doing in the lab, but I'm happy to talk further about anything that comes up, and I'll just stop talking about a quarter after about 45 minutes because I really want to hear from you. A little bit of disclosure. I'm talking to you from the United States with a perspective that is different than yours. So I'm very interested in what your take and perspective is on some of the topics that we're covering. And I apologize if I am uninformed about some of the wonderful knowledge that you have that you can bring to the table. So we all have a lot to learn. Okay, and then, yeah, so I wanted to just take a minute to tell you what I am. So I'm a behavioral neuroscientist, and stop video. And there are people coming in. So I'm multitasking. Let me get these people in. And essentially what that means is I'm interested in looking at the relationships among behavior, like animals. So we use, this is a rat like we use in the lab, a Long Evans rat. So we talk about preclinical models. So a lot of our behavior, we can house these animals in the lab very easily. There are limitations to that, and I'm happy to talk to you about the limitations of just focusing on rodents. But we know a lot about rodents, and then we are interested in how their brain controls or influences their behavior. We're also interested in things like hormones and the environment. So we're going to talk a lot about the environment. We still have some more people coming in. And I see the environment as sculpting our brains. And so hopefully it makes us more accountable for the environments that we put our brains in, as we learn that our brains are always changing. So I don't know why. Here we go. Although we focus mostly on rats, I did mention and would want to point out that it's important to look at other animal models, because it's very limiting to just look at rodents. Rodents are great. So we've looked at a lot of different behaviors with these rats. Maternal behavior is something we've been fascinated with, and paternal behavior, but both of these species are carrying their offspring. Just the mere act of changing your brain, if you're an animal, to take care of a whole nother animal, and when they need to eat and to rest. The brain needed a lot of ammunition and resources to be able to do that. And I think that parenting played a huge role in making us who we are, hopefully caring, sympathetic, even though humans have their moments. So in addition to the rats, I'm very passionate about raccoons. I'll mention them. They're very clever. We have them all over the place in North America. And we do some non-invasive work, just observational work with some primate species that the different species have wonderfully different behaviors. Like these owl monkeys are the only nocturnal primates. So they're active at night, and their big eyes are a clue about that adaptation. The squirrel monkeys that we have that are here, this mom carries this baby that it can be up to 1/3 of her body weight, and she's jumping on tiny little branches. So the body and brain adapt to parental behavior in magnificent ways. Most of the rodent world, for example, rats, are single parent, single mother models. The dads deposit sperm, and then they're not around to take care of the young. But this little rodent called the California deer mouse, this is a male, and they are very paternal. And it's so fascinating that about only 5% of all mammals are paternal. Most of the family strategies in mammals is that the mom takes care of the offspring, sometimes with help. It takes a village to raise young. So we like to look at different species, but in my lab, I have the rats. So don't have a lot of time today, but I want to talk a little bit about the differences and uniqueness of human brains compared to other animal brains, and what this neuroplasticity, this topic of neuroplasticity is, that it's just so fascinating to me. And in order to have optimal functions, we need to be aware of vulnerabilities for mental illness. I know in the US and across the world, depression is, it seems that rates are rising. We're not doing a good job in treating this. So we're going to look at 2 models that we're looking at with rodents, actually, that may provide some insight about building resilience against emotional disorders such as depression. And then enriching our brains. What types of environments and activities can we put our brains in that has been found to be beneficial? So that's a lot, but those are some of the topics that we're interested in. The amazing human brain. As you've learned in your biology classes, we're on a continuum with other animals, certainly primates. So we're not completely different from other animals, but our brains are unique. They're very large for our species. So, and they have this wonderful, we call gyrification, the wrinkles in the outer area, the cortex, that allows us to pack more tissue in this more recently evolved area called the cortex. So we're packing a lot of neurons. The neurons, the brain cells here are densely packed in the human cortex, more so than other animals. So as our brains got bigger, the cells stayed the same size so that we could pack more in. And we think that that probably is related to, well, we think that we're special primate, premier animals, but we do have some pretty impressive problem-solving abilities and such. So we think that this richness of neurons is related certainly to our abilities, our intellectual, cognitive abilities. But just to compare, it's interesting, a colleague of mine at Vanderbilt University, Dr. Herculena Huzel, and I can give you her name, I should put this here. She's come up with a technique, and if you Google brain soup and TED Talk, you will see her amazing talk, where she takes parts of brains and homogenizes them. So she makes soup, a liquid, and then she's able to take out certain volumes and extrapolate the number of cells for that particular brain area or brain. So it's her work that we used to think that the neurons, the cells, one component type of cell in our brain, that the humans had 100 billion neurons. That's in a lot of science fiction movies or sciencey kind of movies. The humans have 100 billion neurons and it's so impressive. Well, no one had really done the empirical work. It was almost like what we call an urban legend. It was an estimate. We call it a guesstimate more than an estimate. But by doing this more systematically in this process that she uses, oh, I do have her name here, apologize for that. So, and she calls it isotropic fractionation. We know that the humans have 86 billion neurons instead of 100 billion. And you may think, 14 billion, does that really matter? Well, that's an entire baboon brain. So that is probably an unacceptable margin of error. But it's so interesting to look at other apes like the gorilla. And the brain is smaller, and you see in our cortex, and this is what this pink is, that outside area I was so excited about, we have 16 billion neurons. The gorilla only has about half of those and much fewer neurons. It seems that the other apes devoted more energy toward their big bodies to defend themselves and such. So that's an interesting difference even among apes, that we're standouts among the apes. We don't have the largest brain. Other animals, dolphins, and certainly elephants have a larger brain. But even with this huge brain that an elephant has with 257 billion compared to our 86 billion, they only have about a third the number of neurons in their cortex. So as Dr. Herculena Huzel wrote in her book, The Human Advantage, we're a little different in that way. And she has very interesting theories. Perhaps cooking was what gave us an opportunity to have more energy devoted to supporting all of these neurons. Well, the rodents that I use aren't as impressive. They still outsmart me every day. But they have closer to 70 million neurons, and million. So we're not talking billion, with 14 million in the cortex. So it's important to understand some of the comparative differences with different brains. So this term neuroplasticity, you've probably heard about that if you're interested in medicine and biology. But this refers to the fact that our brains are changing throughout our lifetime, our entire lives. I like to say that our brains are changing from the womb to the tomb. So if we're living, our brains are changing. Hopefully they're changing in positive ways, but they can certainly change in kind of negative ways as well. So one way, a popular measure or metric of neuroplasticity is this term neurogenesis. And this refers to the creation of new neurons. And we certainly have the creation of new neurons as our brains develop. Starts from scratch, and we get these 86 billion neurons. But once we're born and other animals, there's still evidence of new neurons being produced. It's controversial how long and at what rate this happens with humans, but we certainly have evidence during childhood that new neurons are being produced. And there are other ways to change our brain without investing in the production of entirely new neurons. And this may be the structure you see here. This is an area called the hippocampus in one of our rat brains. This area is involved in learning and memory and spatial kind of navigation of their environment. We used a stain that stains entire neurons, but not all of them, because it would be a black kind of smear here. So for some reason, it selects about 5% so that we can trace and count neurons. And then we stained also, this is a stain, a thionin stain that stains the cell bodies. So you can see how many neurons and nuclei were here. And then we just have some good examples here. But brains look like a beautiful garden. Their organization is just amazing. So we can get new branches. Our branches can fall off. There can be no new areas for connections. We call synapses, where neurochemicals can be shared. So there are more efficient ways of changing our brains than creating entirely new neurons. So even though neurogenesis has gotten a lot of attention. Well, we can train our brains too. I mean, sometimes when I was coming up as a student, a lot of us felt that we were stuck with the brain that we were born with. But through some of the things I'm talking about today, we can certainly change our brain through training. So there's some fun research looking at how juggling, so learning that motor task can increase, in this study here, white matter, its connections among the neurons. It looks pretty far back, but it's in an area, the parietal cortex, that's involved in processing information about the body and space. So research has shown that before people start juggling, they have one level, less than impressive level of white matter here, or the size of a brain area. And after several months of juggling, it pumps up like a muscle. And then if they stop juggling, you have atrophy again. Sometimes in class, I give all my students juggling balls for them to practice, and we don't do brain scans, but I tell them that's probably helping them with their other study endeavors. There's another fascinating area showing that training can change our brains, even in adulthood. In London, I could never do this because I don't have good spatial ability, but the taxi drivers have to take what has been called the hardest test on the planet. It's called the knowledge test. So in order to be a taxi driver, you have to memorize every street in the city of London. So that's 25,000 streets, 60,000 like restaurants and places of interest. So they do not rely, and we rely on navigational systems. We get in our car and just punch it in, and then our brain is just very passive. So they don't have maps, they don't have navigational systems. They have to learn this. So this takes up to 4 years, like the equivalent in some places to be a doctor, to memorize the streets of London. So they have shown that this increases the hippocampus. I just showed you the hippocampus in the rat brain. It becomes larger after these taxi drivers have studied for the knowledge test and passed that. So hobbies, studying, a lot of things that we do is going to change our brains. So we need to be accountable. We'll talk more about that after this section. So another area, as I mentioned in the beginning, that's so important for maintaining healthy brains is to be aware of when mental illness, stress, emotion, anxiety, emotion's good, but some of the emotions that are taxing, such as extreme anxiety and stress, seem to wear down the brain. So we've looked at some animal models of depression in our lab. And so you may think, how do you know if a rat's depressed? And that's a very good question. We don't, and I'm sure rats are not depressed like we are, but we can look at symptoms like not enjoying their sweet water or rewards, not being as social, not exploring their environment, having high stress hormones. So that's the way we can look at this. And we're very interested in just being detectives and trying to figure out what is going on with depression, with the rates rising so much. And we're doing a better job. And you, those of you studying to be doctors, with cardiovascular disease and diabetes and AIDS, and fewer people are dying. And the treatment is better for some of these non-psychiatric illnesses. we don't have that success with depression, and schizophrenia and other anxiety and psychiatric illnesses. We need to do better. So we try to do our little part. Now in the US, and I'm interested in treatment approaches where you are, but we're very dependent on antidepressant, on medications, on the pharmacological approach. This has been related most closely to serotonin, with the idea that if we're depressed, we have low levels of serotonin. And if we take a drug that increases momentarily that neurochemical and some others maybe, that will make us feel better. But it's correlational research. If you go to the doctor here and you're prescribed an antidepressant, it's not because the doctor did a test to show that you have low serotonin levels. Unlike if it's suspected that you have diabetes, your doctor is going to look at glucose and insulin levels. So we don't have that specificity. It's just the idea that this may help. And it is not as helpful as it should be, in my opinion. With some studies, it doesn't do much better than the placebo. And at least I think we need to look at adding some treatments to this line of therapy, because we have not done as well in treating. And through COVID, the rates just are increasing. And interestingly, if we're detectives, it's interesting that, and I think across the board and across the world, women are twice as susceptible to depression. So there may be some sex hormone or something related to reproduction or something culturally related to sex that is important. So we want to look at all the clues. But several years ago, I was frustrated. I was writing a textbook about depression and said, we need better treatments. And so I wanted to just go back to the drawing board, the brain board, to look at what brain areas were kind of aligned with the symptoms that we associate with depression. And what I came across is, and I kind of need to back up, is that a lot of the brain, as we just look at the territory of the brain before I get to the symptoms of depression, a lot of the brain is devoted to movement. So this cerebellum that hangs off the back of our brain that the elephant has so many neurons in, it has most of our neurons, like 78% of our neurons are in the cerebellum. It's involved in motor coordination, movement, not solving physics problems, but most of our neurons are in this area that's involved in motor coordination and some other things like conscious awareness. Also, this big middle part of our brain, the striatum, is involved in movement and the fluidity of our movement. People diagnosed with Parkinson's disease or Huntington's disease, where the movement is jerky and it's not fluid and it's hard to initiate that movement, that's a disease of this striatum. And we have this big strip right here around our ears that coordinates our voluntary movement. So I'm aware of me, I'm gesturing a lot, even though you can't see that because it seems to facilitate my language. So a lot of our brain is devoted to movement. And I was wondering if that was related to disrupting our brains, especially considering that our movement has changed over, at least and over here, over the last century. We've become more tolerant of sitting in front of screens and not being very active. And when you look at brain areas, which is where I was starting with before, one symptom of depression is this lack of pleasure. And the nucleus accumbens, I'm oversimplifying, but it's involved in telling the brain when something is important. And this is an area that is activated with drugs of abuse. So someone taking drugs, it's influencing this brain area. It's very rewarding. It's not good for the brain. It kind of hijacks the brain when it's something artificial like drug use, but it's very good to direct us to food and social interactions and training and achievement. So this talks to the striatum. There's a pathway that goes from the nucleus accumbens to the striatum and also interacts with this prefrontal cortex that's involved in more of our executive cognitive functions. And so whereas most theories about depression were about emotions and cognition, and that's certainly important, I started to think more about movement. And as we're moving our bodies less and our brain is kind of downsizing to adapt to me sitting in this chair for way too many hours every day, does that have a downsizing kind of effect on our brain that could lead to depression? It's so interesting to think. Now, in the US in the 1930s, the World's Fair came to the US, and they introduced this brand new impressive technology called the television. And no one had seen anything like it. It was interesting and intriguing, but the New York Times, which is a pretty reputable news source, I guess, said that, you know what, people don't have time to sit in front of a screen and watch something. We're really busy. So for that reason, the radio is going to be more popular, because you can work in your kitchen or carve your wood or fold your clothes or whatever you're doing and listen to the radio, maybe like we do a podcast. But this TV thing, sitting in front of screens, that would never go over. Boy, were they wrong, and boy, have we changed over the last century. So as we have traded in our spears for selfie sticks and been all too happy to sit in front of screens, I do wonder what effect that has had on our brains. And sometimes I talk about prehistoric Prozac. Our ancestors may have been a lot more active, physically active, that could have facilitated that neurochemical like serotonin and other neurochemicals that kept them from being depressed. That's just theoretical kind of just supposing something like that. So this idea that our nucleus accumbens can form connections with this middle part of our brain involved in movement, and those connections can motivate us to engage in these behaviors, became something that we were interested in, and I wanted to take it to the lab. So we're back to our depressed rats. But generally, what I came up with is, with my students, is letting more people in here, this idea that I wanted rats to go out and work for their rewards. And so we kind of cheating and using some sweet cereal called Fruit Loops. And it's not very natural, so perhaps I shouldn't do that, but they like it. It's crunchy and it's easy to give to them. So one group has to dig up their rewards, and they get all the food they need, so they're not hungry rats. This is just their treat. So 5 to 10 minutes a day, they get to go out to the fields and dig up about 4 Fruit Loops. The non-contingent control group gets the same number of little Fruit Loop pieces, but they don't have to work for them. So they're not contingent on their behavior as these animals. So if we're going to be kind of cutesy, so our contingent animals, my students, we call the worker rats, and the non-contingent, the trust fund rats. And we're interested in what impact that has on their resilience and persistence. And we've done lots of studies, but just to give you a few highlights, when we give these animals another challenge test, and this is not very sophisticated, it's just a cat ball and it has a Fruit Loop in it, but they can't get it out. And the ones who are the contingent worker rats, they persist longer in trying to solve the problem before they give up. And we think that's resilient. You may argue otherwise, but it's as if my daughters say, "You've got this, Mom," or they say to each other on a team, "You've got this." It's as if they're thinking, "I've got this." And it makes sense. Decades ago, when cake mixes in this country came out, they wanted to make things easier for women, where they just put everything in a box and just add water and you have your cake. But they realized that that wasn't enough effort for mostly women who were doing this. And if they just took out the egg and had them add the egg, that it gave them more pleasure and pride. So they were very smart to have the women or the bakers work a little bit harder, so they would be proud of the outcome. So it seems to this training and having to work makes you a little bit more persistent. We've also looked at stress hormones, cortisol and humans, but corticosterone is a version in our rats. But if our brain perceives something as stressful, areas of our brain, such as the hypothalamus, tell this endocrine gland to release a chemical, ACTH, that goes to a little gland on top of our kidneys, of all things. And that releases some hormones, steroid hormones, such as cortisol, the traditionally known stress hormone. And we've been very interested in, we'll just call it DHEA, that may serve as a buffer against the toxicity of chronic stress. Stress is wonderful to get us away from a threat, an immediate threat. Not so wonderful when it's chronic, like living through a pandemic or extreme loss or grief. So we've been interested in looking at this in our rats. And one way that my colleagues have used as a depression test for rats is you put them in water, and if they float, they think they've given up and they have conditioned despair. That's a big word term for a rat. And I think it's adaptive to float though. So once you figure out you can't escape, it makes a lot of sense to conserve energy. And if we look at, so we'll look at floating in a minute, but when we swim the animals 3 times, so these are our trust fund rats and these are our worker rats, and we look at the ratio between DHEA, that healthy hormone, and stress, the higher being the healthier, it seems like the contingent animals are getting more resilient with higher ratios as they have more experience with this challenge. And the non-contingent animals are not changing. They're not becoming more resilient. So we're very intrigued by that. And also when we have chronic stress, unpredictable stress that may last for about 2 weeks, where every once in a while we expose them to like a cat odor or a swim, it's those trained worker rats that have the higher ratio. They get better midway and higher at the end. And there was no change, no adaptation in the animals that did not have that effort, we call it effort-based reward experience. So I'm talking way too much, but we were also looking at neurotrophic factors, which is a measure of neuroplasticity. And I'll bring this up quickly. This brain area right under the hippocampus that I'm fascinated with right now, called the lateral habenula. So this is the medial habenula here, and this is the lateral habenula. I thought I had, oops. Sorry. Yeah, I skipped a slide. So the lateral habenula, if an animal like here is, there's a bird of prey, they may freeze, especially when they're in a small space, so that that bird won't see them. And that freeze response, you can see there's some similarity to something like depression, where you're not making an effort, you have kind of lethargic, that you're not very active, you're more passive. And as I'll mention, a new drug that has received a lot of excitement for depression is ketamine. That's really an anesthetic drug, so you need to take it very seriously. But it seems to work via the lateral habenula. So this area that may tell the animal to freeze, to be passive, could be involved in depression. And ketamine does work via that area. And you can see all the connections that this area has. Letting people in. So we found that our trained animals, when we look at the lateral habenula, well, let's just say our trust fund animals, have more neuroplasticity in the lateral habenula. That suggests that maybe they're more, have a higher probability of freezing, that there's more activity in that area involved in passivity and freezing, which is very adaptive in some circumstances, but not for long periods of time. And when we look at an area of the hippocampus, we don't see that. So we're looking at neurochemicals and hormones and behavior, and that's the, I'm sorry, my computer is not working very well here. I came up with this term and it's not given the whole picture here, but behaviorceuticals, just kind of made that up, that we can change the neurochemistry in our brains with our behavior. For example, knitting, the repetitive behavior can change our serotonin that we talked about as being important, maybe as an antidepressant. Dopamine, as we look forward to seeing that the end result of our knitting or activity, and that works in that nucleus accumbens area that I talked about, that could be involved. And stress hormones, it could reduce the stress hormones, because I should have mentioned that about half of people who are diagnosed with depression have high stress hormones. So it's certainly something we need to be aware of. And if we knit with a group of people, that can increase our oxytocin, which is known as the cuddle chemical, oversimplifying, but that can also reduce our stress hormones, and it feels really good. So behaviorceuticals with effort-based rewards. So we need to be careful about how easy our life has become. If we take all the effort out, we may be downsizing our brains. I mentioned the raccoon, and they've got paws like our hands, and they have these squiggles on their brains like we do, and wonderfully complex brains. So we just published our first paper with raccoons, and looking at problem solving and starting to look at hormones and coping strategies and such. So more to come there, but just an example that we need to look at more and a diverse array of animals, and also look at animals out in the wild. And we're trapping wild rats now, and that's been very interesting. Another type of effort that's important is exercise. That's different. You're not seeing a product that's being produced, but it's still getting oxygen and glucose to our brains. So that's important to exercise, and exercise with rats and running wheels at least does increase that neurotrophic factor in the good areas of the brain, hippocampus and such. So it's been correlated, exercise, probably nothing new to you, has been correlated with neuroplasticity and a lot of positive emotional and cognitive effects and other general health effects. We're also looking at coping strategies, and the take-home point here is that when we profile rats by doing this little back test and do it more than once, the animals that are flexible, like one day they'll be active and another passive, as if they're learning that they can change their responses. This was based on some Swedish work with pigs. It looks like they're getting a massage or something, but you can look at the kind of a personality difference. So being flexible is where we're seeing animals that change their behavior, like in this float task that I think it's more adaptive to float. Our flexible animals change from, so this is time floating, they start to float more and sooner than the other animals, as if they're adapting more readily. So your coping strategies are incredibly important. And also the environment that we put ourselves in, I'm going to call it an enriched or an engaged environment, is really important. We can change these environments with our rats in what we call an enriched, as I mentioned, an enriched environment. So we change kind of like toys. It's kind of like a Disneyland over here. Every third day or so, we put new elements in their environment, and they have a bigger space so they can move around and they can interact with each other. Unfortunately, this is the traditional way to house animals in laboratories across the world, really. They do have food and water, so this is a little bit atypical, but certainly in my lab they do. But they don't have, it's very restrictive, and I worry about the generalization and the authenticity of the data we get by looking at selectively bred animals in these very restricted environments. We've looked at kind of artificial environments and natural environments where we bring in dirt and plants. It's autoclaved and safe for the animals, and wood and straw, same number of elements. And this seems to make the animals a little bit more emotional resilient. They're both equal in their cognition, but these are more emotionally resilient. And this makes sense in light of some fascinating research in Sweden, where they have found that people who grow up with green stuff around them, like trees or a park, are about 50% less likely to be diagnosed with a psychiatric illness. And so, as Professor Dajani was saying, green space and nature can have a huge impact on our brains. Our brains evolved to be in this setting more so than this setting, but a lot of us are in this setting. So I think that what we're learning from what we call our city and our country rats is translating to what we're seeing in humans. So we should use those models to learn more about them. Most labs are not going to have those big, messy environments. So we played around with in-between type of environment. We call these the University of Richmond rat lofts, where they can go upstairs or downstairs and just have that movement to be able to leave and come back. Seems to be very important for a mammalian brain. And it's so funny to see them scooting up like they're in little subway cars or something. And as we're testing them, these guys are testing similarly to the other more traditional enriched environments. So way off course, still talking about enriched environments, but a very different task. We use some old-fashioned psychology, Skinnerian conditioning, operant conditioning, to train rats to do a very complex task. This was drive a car. Humans drive a car. I know they don't drive a car, and I'm talking about nature, but we learn tasks as well. So this was our first kind of prototype of a car, and we would train them to go in to get a Fruit Loop and to press the bar to close the loop to drive. And then this is our third kind of version of our rat cars. They've become quite sophisticated, working with robotics. We had some film crew come out, and we had an actual rat race that we talk about where they raced. And so we actually taught them to steer and to go toward a goal. But how this is related to what I was just talking about, we have found that enriched animals, they learn the task. What we saw was in 22 trials, whereas the standard housed animals, well, in that study we did, they never learned. So our enriched animals got their driving license here in the US, and the standards did not. So they still need to work on it. So our environments are important. And there's so many questions we can ask about brains. And I think all the time, what is the function of our brain? Obviously, it keeps us alive with our glandular secretions and muscle tone, but it also, we're gathering information to be able to make decisions. Do we want to be a doctor or a teacher or a professor or have children or get a pet or what toothpaste to buy? We're constantly making decisions. And the more interactions and input we have from our environment and our experiences, it builds that cognitive capital that I had in that other slide. It also helps us to sense, and this is some of our more current work, we call it pattern separation. When something is just subtly different in our environment, we need to notice that. It could be danger, it could be an opportunity. So our trained rats with the effort-based rewards, they can notice changes in the environment. These are very similar, but when there's the very subtle change, they see that more readily. And that's a healthy brain too. We want to be vigilant. We want to see when there's danger, when there's an opportunity. So they look like they're at a art exhibit here, but when they go to the one that's novel, when they've been exposed to two that are exactly the same, that suggests that they're recognizing that it's different, that they're sensitive to their environment. So I'm 1 minute over, but our takeaways, our brain, the human, we're part of a continuum with other mammals, but the human brain seems to be unique in some ways, especially with the dense number of neurons in our cortex, and this changes from the womb to the tomb. Our experiences sculpt our anatomy and our neurochemistry. This makes us accountable for our neural health. Some of us have more agency and flexibility than others, but it's important to understand that. Environmental, social enrichment and engagement are important for building our stockpiles of neural networks so that we can make optimal decisions and informed decisions. And relevant and effective training, again, can change behavior and optimize our performance. Research is not done on my own. This was this summer, we had lots of students that were working in my lab and a visitor, Dr. Kent, who brought a few students as well for a cookout. And everybody works really hard, especially during the summer. So I thank my students. I love when they put that white coat on, as you will do as you continue your medical education. And it gives them a sense of pride. And they are my partners in crime and neural and behavioral neuroscience detectives. I appreciate their work. I was just going to mention, if you are interested in some of these quirky stories, I have a YouTube. If you just type Google YouTube and the Brain Chronicles, I have 5 mini lectures using something called the light board, which is a new storytelling technique about maternal behavior and behaviorceuticals and movement and some other cognitive decision making. So feel free to check that out. And I am done with being a few minutes over. So I will stop the share so maybe I can see your faces.
Thank you so much, Professor Lambert. That was amazing. What I love about your work is how you tie the basic sciences and the theories into something practical that we can, sit and reflect upon ourselves and our daily activities. What can we learn from basic science?
I hope so.
To make our lives better. So thank you.
Oh.
I mean, that knitting during COVID, I made 4 blankets. So
that was
a good therapy.
You're so smart. Yeah. It's great.
So thank you for that gratification. And it speaks to the past, right? To our grandparents and older generations, how they were much healthier.
Absolutely. Well, they, every era has its challenges, but they certainly moved their bodies more and had more sense of agency and control over their lives because they understood, I don't know how to repair my computer or car or anything, but our grandparents repaired everything and they were more resourceful. You're right. So.
Absolutely. So let's turn it over to the audience. We have some questions in the chat. Can you see those, Kelly?
Okay.
Can you see your chat? And maybe you want me to read it or you'd rather read it? Which would you
see? So let me look at Lena. So what if we prove something or discover a finding in these animals? How can we prove that it also applies to humans, even though we will face many ethical issues regarding testing on humans, and especially tests involving brains and nervous system? Also, how come we still haven't found a cure for neurological diseases such as Parkinson's? Finally, it's an oxytocin, the hormone released during positive feedback when females give birth, so it helps reduce stress. Yeah. So this person has her PhD in neuroscience. There are many challenges to using these preclinical models, and I think we need to be cautious and note those limitations. So I think that we can learn some fundamental information about what a neuron looks like and how neurons operate, but we're limited in translating to humans from the rodents. And I think that's why we don't have cures, especially for the for the psychiatric illnesses, but even the neurological illnesses like Parkinson's disease, where we know that that's a reduction in dopamine, but we still haven't come up with the right treatment. We have pharmacological, surgical, fetal cell transplant, and everything is trying a lot of different therapies. So this is something we struggle with, the ethics that's involved with doing animal work. That's a personal, I always talk to my students about, we follow all the regulations, but still it may be a personal perspective not to use animals, and whether you value humans above those animals is important. So that's a very deep process and thought question that I respect, and thank you for asking that. Why haven't we found cures? We need to do better. You're right. And it's unacceptable. And that's why we're trapping wild rats and we're trying to get raccoons. And we can do a lot of ethical work with humans with non-invasive biomarkers and doing post-mortem work. We have brain banks in this country where you can get some tissue from people who maybe died by suicide or had Parkinson's disease and look at the effectiveness of treatments or cause or susceptibility. So we need to do better. And yes, oxytocin with maternal behavior, that is involved with giving birth and lactation and forming relationships. And again, parenting kind of paved the way for human interactions and the complexity of the brain. So lots of good stuff.
Thank you, doctor. Thank you, doctor. It means a lot. Thank you.
Oh, that's a great question. Okay, let's see. So is there another?
No, I don't
okay,
think so. I mean, maybe, so please, anyone who has a question, you can just unmute yourself and ask, and any question is fine. There's no simple question. There's no silly question. Every question matters. If you have it in your mind, then it's important.
Right.
And probably somebody else has it and they feel they're holding back not to ask. But so please take this opportunity and ask Professor Lambert anything that occurs to you or something you didn't understand that she could clarify as well.
And I'm interested in Jordan, what are the attitudes and responses about different types of treatments for depression, and how readily are people to, how do they seek help? Is there stigma more so than maybe other places? Someone is asking a question, so that's something throwing out there. Is it possible that experiments on lab rats get improved by making rats experience more similar environment to humans so the results are more accurate? It's perhaps we can get into this question. It's a very good question. Are we wild or domesticated? I think about this a lot. So it could be that we're more domesticated and maybe we're more like those standard rats and those stripped down cages. I think we have more engagement and unpredictability in our world, but I'm working on a book now about wild brains, or a proposal, And I think out of convenience, we've bred laboratory animals to be very docile, easy to handle, not very stressed and aggressive. And the stress response is important in understanding about psychiatric illness. I said half of people diagnosed with depression have high stress hormones. So we may have bred out some of the critical areas of the lab animals that we need to do a better job at developing interventions. So you're right, it may be, and the raccoons, they live in our attics, and they live in our cities, and rats that are wild live in our cities. So all of those species are very opportunistic and adaptive, but when you put them in a situation where they're all kind of trust fund rats, we just give them their food, they don't have to work for it, they don't have to search for it, we have changed them. And so we need to be careful and vigilant and not just get so caught up in, I'm doing this fancy research and I have this grant, but maybe we need to look at some different species, and maybe we need to look in the wild to confirm and validate, like the swim task. It's very controversial. That may not be a test of depression, but there's a lot of studies that have used that as the gold standard. If the rats keep swimming, I've got a drug for depression. I'm a little bit skeptical about that. And we all need to be skeptical. I hope you're skeptical and you ask questions, and we need to be critical of this, but good question.
Oh, here we go. We have another question here.
Okay, I believe that there is still stigma, and this is related to my question, that it's kind of unacceptable to seek help from a psychiatrist. However, awareness is being raised and more people are seeking help nowadays, and more people are talking about it and sharing. That's good. I know COVID, the rates have increased. And what I should have said, my effort-based reward model is kind of an animal model of something we call cognitive behavioral therapy in the US. So there are models where you take drugs, and there are models where you see a psychiatrist or a therapist and you talk through how you change your goals and behaviors to try to change your anxiety and brain. And sometimes it's combined with medication. And so I'm trying to learn more about that cognitive behavioral therapy, because in this country, we think of medicine as being a pill or surgery, and we don't appreciate lifestyle changes and some of these more holistic approaches as being scientific and evidence-based. So that's why, we're trying to take some kind of cognitive behavioral therapy and test it to see if the outcome is similar to antidepressants or ketamine that a lot of people are excited about, that I hope we're very cautious with in this country. So there are different approaches. Exercise, having a hobby. We need to look seriously at all this and how it changes. So another question, is it just the density of the nerve cells in the cortex that makes us different, or some kind of special bonds or something that differs between us and other species? Good question, and I recommend reading Dr. Huzel's The Human Advantage. So we do seem to have, more neurons in our cortex than any other species on the planet. But there's also, we need to look, there's a type of cell that are in some mammals, and we also identified it in the raccoon. So it's not unique to primates, but it's a long neuron called a von Economo neuron. It's also referred to as a spindle neuron. And we think that those are fast projecting cells. So when we see something, when there's a threat, like there's a car accident or something, you can feel there's been a response before you actually process it. Or you touch a hot stove and you pull your hands back before your brain knows what's going on. So those fast projections related to reflexive fast responses, but also making connections and being able to make decisions kind of, as we say, on the fly or quicker than the other person so that you can survive. So having more, Susanna uses an example of having a pile of Lego blocks. It's not enough just to have those blocks. So these connections that you mentioned are important. But through experience, building these blocks into a city or a building or a castle, that's what our brain is doing. And so with more neurons, we've got more connections, but more things can go wrong too. And we're totally dependent with this neuroplasticity on other people. If you drop a human baby on an island, that baby's not going to survive, whereas other animals may. They're different strategies. So we have a huge vulnerability in that we are dependent on others. We're very social in that way, but we have brains that are very flexible. So if we can get together with other brains, we have this huge mega brain, and that's what a community is, right? So some people are good at different things. So something emerges that's much more powerful with multiple brains than single brains if we can behave ourselves and keep things in mind. So I went all over the place with that, but a lot to think about. I heard every generation gains 3 intelligent points and closer to ADHD behavior. Does it affect the brain ability to gain more information in early age and getting depressed more often? Wow, it's really interesting. It's controversial, these intelligence tests, and I'm more of an animal behavior person, so I'm going to be a little bit more skeptical. In some ways, we may be, we're testing things that we think are intelligent, like physics problems or vocabulary. I'm not sure vocabulary was incredibly important for the survival of our ancestors, but flexible thinking, cognitive flexibility, problem solving, being able to change and adapt, that's what I think about. So yes, we're changing the way our ancestors learned. We're learning a lot, like that taxi driver test, the knowledge, that's almost ridiculous how much expertise and knowledge you get with that. Does it have some impact down the line? So yes, there's some research that we're doing better on some of these intelligence tests, but maybe we're kind of teaching toward the test, and there could be some confusion or controversy about what those tests are actually testing. But what we're measuring as intelligence, we do seem to be getting better. And ADHD, we're creating a world that is so different than our ancestors. You see me trying to let people in and talk and do this, and I'm not doing a very good job of this, but, and we're not great multitaskers, even though we like to think we are. We're not. We still need that focused attention if we want to solve the problems of the world. So we have so many things competing for our attention, and if we don't pay attention, attention is the wonderful resource, it's a diminishing resource of our brain. We're going to be limited. I catch myself all the time when I park my car. I don't pay attention where I park. I just get out and I never know where I park. So I have to be vigilant about, okay, pay attention to where you're parking, because I'm on to the next thing as soon as I get out the car. So we can be more vigilant about paying attention and appreciate that we need to pay attention. And it's not so impressive to be able to multitask and do 20 things at a time. We're probably going to make a mistake. I don't want, if you're going to be surgeons or whatever, I really don't want my surgeon doing 20 things at a time. I want focused attention, and the brain still requires that. But flexibility and challenges are good as well. Such great questions. You have such a great class.
Yeah. Oh, thank you. No, I mean, it's so fascinating, and they're fascinated by what you're saying, and they're fantastic students. Absolutely. So we're at the top of the hour, and I just want to, a take-home message, I think, that I really see powerful is that that flexibility, that plasticity, that we're not born as we are. So much can happen, and by exposing ourselves, by stimulating, by doing things, we can have some control over our lives to make them better.
And hope.
And hope, exactly. Being positive and optimistic while acknowledging what we have around us. So I think that's a powerful message. And we're all excited to watch your videos, read your books, and follow your research. And this is a call my students to think that Kelly said that she likes to hear other perspectives because that enriches her research.
Right.
And so please reach out to Professor Lambert. You never know, you may go up and spend a summer in her lab and do some research.
Hey, Lambert at richmond.edu. Saw that on but I'll send the talk too. Yeah,
we'll send the talk, and I'll share her email and her books and her links to anyone who wants to follow up. But thank you so much, Kelly, for your time and sharing. It's always a delight to listen to you.
Yeah, Dr. Dajani's my science soul sister here, but we need a generation of insightful, inquisitive, evidence-based researchers to try to solve some of these problems, because my generation is not doing very well. So we need you guys to do a better job. So I hope you have a wonderful, it's night there, so rest of your evening, and thank you for taking time at an odd time to hear about rats and raccoons and brains and all my quirky topics.
No, thank you. Now we have much more respect for the rats we see in the street.
They're smart.
Yeah,
they're smart.
Exactly.
All right. Well, I'm going to sign off, and I will send you a link to the recording. I have an appointment now, but I'll send that later tonight.
No problem. Thank you very much. Have a great day, and everybody, good night. Bye-bye. Bye-bye.